This video is on DNA structure and replication. So for life to exist, a cell has to be able to replicate itself. And in 1953, James Watson and Francis Crick, who are depicted at the bottom of the picture here, determined the three-dimensional structure of DNA. And they actually got the Nobel Prize for this. The top picture is Rosalind Franklin, and she also contributed to this, but she was not eligible for the prize because she was not alive at the time. But this was in 1953, so this is a fairly recent discovery, all things considered. So DNA is a two polynucleotide strand that are twisted around each other, forming what's called a double helix. Each consists of millions of nucleotides. So each of these helixes consists of millions of nucleotides. Your monosaccharide phosphate backbone is on the outer part of this. And the two strands that make up this double helix, we say they're anti-parallel, which means that they are oriented oppositely. So if one is 5' to 3', the other one is oriented from 3' to 5'. And we know DNA stands for deoxyribonucleic acid. So in this structure here, you can see your bases, nitrogenous bases. You can see that these bases are these horizontal colored bars. So this red and purple bar over here, those are your bases. Your sugar phosphate backbone, that is the brown ribbon in which you see right here. And the dashes that you see in this structure, these are your hydrogen bonds. So your dashed lines are your hydrogen bonds. Notice that some of them have 2 bars and some have 3 bars. And we will talk about that on a later slide. Also notice how they're oriented anti-parallel, 3 to 5, and 5 to 3. So Rosalind Franklin, who didn't win that Nobel Prize, hypothesized by looking at this structure, this X-ray, she saw a lot of symmetry in it, as you can see here, and she hypothesized this helical structure of DNA, and it turns out that she was correct. So this is an X-ray showing this. So your sugar phosphate backbone, this is on the outside of the helix, and this is in an aqueous environment. That's why it's hydrophilic, water-loving. Your non-polar, your less polar nitrogenous base, that would be on the inside for the opposite reason. These bases form these hydrogen bonds. Again, it can be 2 or 3, as we saw in the picture, to complementary, it's called a complementary nitrogenous base on the other strand. And that is called a base pair. So these are very important. A complementary nitrogenous base or base pairs is when you switch letters. And these letters are switched from A to T or T to A, and then G to C or C to G. And we will practice doing this shortly. And these A's and T's, remember, that stands for adenine or thymine or guanine or cytosine, and we saw that in a previous video. Now, your 2 lines for hydrogen bonding, they are for your A's and T's. So in that previous slide, the 2 bonds, that's A to T. Your 3 hydrogen bonds, that's for G, not T, that's for G and C. Something called base stacking. This is stabilizing interaction occurring between delocalized electrons of heterocyclic rings of nitrogenous bases. So pretty much your base pairs and your base stacking together, that's what holds that double helix of DNA together. We know hydrogen bonding is not the strongest, but with the base pair and the base stacking together, that's what holds them together. Okay, so again, looking at this picture here, here are your 3s for your G's and C's, and here are your 2s for your A's and T's. So it says below are sequences of nucleotides located on a segment of one strand of DNA cited from the 5' to the 3' end. Indicate the complementary sequence, or the base pair, of nucleotides on the adjacent DNA strand from 3 to 5. So remember, A and T goes together, and G and C go together. So wherever you see a T for its complementary sequence, you're putting an A, so on and so forth. So for this down here, instead of 5', we're doing 3' because they're anti-parallel. And I see a T, I'm putting an A. T, A. When I see a G, I'm putting a C. If I see a C, I'm putting a G. If I see an A, I'm putting a T. And then it ends at 5'. So that is your complementary sequence. And then for the bottom one, same idea here, 3', and then we're just doing TA, CGGT, 5'. And that is your answer for that. So complementary sequences or base pairs are not too difficult to do. Okay, getting into chromosomes and the human genome. So your DNA, if it was stretched out into just a straight line, it would measure about 1 meter long, a little bit more than 1 meter long. But it's not. Our DNA and our cells are actually coiled into what's called chromosomes, which you can see are very small, 1400 nanometers wide. The double helix, which the DNA forms, is about 2 nanometers wide. And this double helix is coiled around histone proteins. And these histone proteins form what's called a nucleosome. These histones are positively charged. So they're going to want to interact with something negatively charged, and luckily the phosphate groups are negatively charged. Once you get these nucleosomes here, they will coil even further to form larger fiber structures of chromosomes. So we will see all of that right here. So your DNA down here, there's your double helix there, the brown structure, and it's going to coil around these histone proteins. So it's going to wrap around these green spheres here, as you can see it doing here. Once it wraps around them, it is called a nucleosome. And then these nucleosomes, you can see they're coiling over here. And what this looks like, here is your X chromosome here. So we know we have X and Y chromosomes. So there is an X one. So it's pretty neat. Okay, another vocab word, something called a genome. So the genome is the complete sequence of nucleotides in an organism's DNA. This differs according to the species that you're looking at. So if you're looking at a bacterium, for example, this has 600,000 base pairs in a single chromosome. Sounds like a lot. Human genomes, we have 3 billion base pairs in 46 chromosomes. So we're talking about a lot of different base pairs here. In humans, 99.9% of the genome is the same for all people. And 50% of human proteins are similar to ones made by other organisms. If we are looking at our genes, gene is just a segment of DNA that contains instructions for assembling a protein. That's probably worth writing down. A gene is a segment of DNA that contains instructions for assembling a protein. Roughly 2% of the human genome contains sequences of nucleotides that code for a protein. And the other 98% have other functions, which some we don't even know yet. We have roughly 30,000 genes, and the average gene is about 3,000 base pairs. So again, you're talking about a lot of base pairs here. Here's a nucleotide down here. So you have your sugar, nitrogenous base, your phosphate ester group. Here is the sequence. Here is a gene. That is where the gene would be located on the DNA double helix, and there is your chromosome. And we can track all of this, which is just wild. And we did this with the Human Genome Project, which is coming up here. So your Human Genome Project was a really big undertaking in 2003. This was a 13-year international effort, and it was completed, again, 2003, so not that long ago. And this mapped out the entire nucleotide sequence and location of all of the genes in the human genome. And this helped us greatly with many different advances in technology, such as genetic modifications. These genetic modifications, these GMs we call them, they're used in foods to get better crops, bigger crops. They're used in medicines. They're used in vaccines. They're used in spraying fields for pesticides, a bunch of different things. So that's gene mapping, really important. This picture here is just showing chromosome number 6. And it's showing all of the different traits and disorders that are associated with chromosome number 6. So you can see there's a bunch of things here. Schizophrenia is highlighted there, breast cancer, diabetes. I think Parkinson's disease is here somewhere. So a lot of different traits and disorders with just one single chromosome. But being able to map it out, we can help better treat it then. Is a histone protein positively charged or negatively charged? And then explain how the charge on a histone facilitates the formation of nucleosomes. So we know that histones are positively charged, and they are attracted to the negative charge on the DNA molecule. And that just makes it easier to coil the DNA on itself and then form that compact structure of a nucleosome. Again, if this was not coiled, this would be over 1 meter long. So it's a good thing that it can be coiled. Okay, DNA replication. How does this work? This starts with the unwinding of a section of the double helix. And an enzyme helps us with this helicase. And what this does is this exposes the nitrogenous bases of the two strands. And we call this the parent strand. These parent strands are the mold, or these are the templates for new daughter strands to come into play here. Each newly formed DNA, double-stranded molecule, has a parent strand and a daughter strand. So they will go hand in hand together. So here you can see your parent strands are in brown and your daughter strands are in blue. You can see something called a replication fork, and we will talk about that. Look how it's oppositely anti-parallel, right? Your 5' and your 3'. So an important enzyme, DNA polymerase, this catalyzes the formation of a monophosphate diester linkage between the 5' ester of a single nucleotide and the 3' hydroxyl group of the growing nucleotide chain. So that's something we saw in a previous video that this enzyme helps with. And this has to be in the 5', 3' direction. It can only synthesize it if it's in that direction. So this replication fork, which we saw before, this is where the DNA separates into two parent strands. So we know these parent strands are anti-parallel. One is 5 to 3, one is 3 to 5. We have two different strands. We have a leading strand and a lagging strand. Your leading strand leads the way, so it is continuously synthesized. So here's a picture here. So here's our leading strand, and you should know that this is assembled continuously. So as this approaches this replication fork here, the enzyme further unwinds the two DNA parent strands. So look at your parent strands in brown, that as you're going this way, it's unwinding, right? It's unwinding here. And the replication fork is going to move in the direction of the synthesis of the leading strand. So since the leading strand is moving down and to the left here, that's where the replication fork is going to move here. So you can see what I'm highlighting in red now, that the replication fork is going to move this way because that's the way of the leading strand is moving. So it's almost like you're peeling back a Band-Aid and the direction fork keeps moving. And that's why it's continuously assembled, because the fork is just going to keep moving for it. Your lagging strand, on the other hand, that is synthesized in fragments. So that is not a continuous assembly. And that will eventually be joined by something called the DNA ligase. And again, this happens in the 5' to 3' direction. So we look up here again, we have our 5' and our 3'. Has to be in that direction. And that's that DNA polymerase enzyme. This is crazy fast and crazy accurate. So it can form bonds between 100 nucleotides per second. And look at the accuracy. Less than 1 out of 10,000 nucleotides is inserted wrong. and it proofreads daughter strands, and it tells itself to fix itself, and then the error rate is even lower, 1 in a billion nucleotides. Now, your mistakes still can happen with this, which is why we do have some mutations in our DNA. So it is not perfect, but it is very good and very fast. Okay, a question here. Which of the following illustrations accurately represents the direction in which the leading strand and the lagging strand are synthesized during DNA replication? So I am looking at my fork, my replication fork, and in all of them, it's here. And I know I want to move from 5' to 3'. I know that's a big part of this. So if I look at, and it's towards the replication, right? I'm moving towards the replication fork. So if I look at A, this arrow is pointed away. It's going that way. So I know that can't be right. If I look at B, I do notice that the arrow is going in, which is good. And it is going from 5 to 3, which is good. So that would be nice for the leading strand. Now I want to look at the lagging strand. This is fragmented. Notice you have an arrow there and an arrow there, so that looks nice. But notice it's going from 3 to 5. We do not want that. If I look at C, okay, my leading strand is from 5 to 3, and it's going towards the replication fork. And my lagging strand is fragmented, and that's going from 5. The arrows are going to 3, which is good. So C looks good to me. And then just checking B, that's moving from 3 to 5. That is not good. So your answer there is C. Using the illustration from the previous exercise, label the following. So our leading strand, we know, is right there. Our lagging strand would be those two. Our replication fork is that area there. Direction that the replication fork moves, left or right. Remember, it's going to move in the direction that the leading strand is moving. So our leading strand is moving this way, so the replication fork is going to move that way, to the left. Sorry, that should be letter D. For E, your parent strands, that is a parent strand, and that is a parent strand. The other two are your daughter strands. So your daughter strands, that and that. Those two are your daughter strands.